In this video, we're going to cover, they're called growth requirements. There are all different categories of organisms and their needs for oxygen and food and all the factors that affect how they produce energy and the names associated with each. So once again, I'm trying to do these as an overview. I'll quickly go through them. Obviously, every cell needs nutrients. And so this is kind of referring to what categories of microbes and how do they acquire those nutrients. In other words, there's a category of microorganisms that are called obligate aerobes. Now that term obligate means strict. So these are organisms that require oxygen. Human beings, we're not a microbe, but we are an obligate aerobe. If you cut us off from oxygen, we don't last very long. So that's a category of microbes. And if you're going to, as a microbiologist, you're trying to identify organisms, you usually look at the, you do some staining first. You figure out cell shape arrangements. You then do some experiments relating to how do they grow. Do they require oxygen? Do they not? The thing is, the bad microbes, the really pathogenic ones, they're usually obligate aerobes. The ones that cause like tetanus, gangrene, those are anaerobes. And an obligate anaerobe, oxygen kills them. So they use fermentation to actually convert energy. Facultative anaerobes are like a switch hitter in baseball, people who can bat right and left-handed. They can use oxygen if it's there, or if it's not there, then they can produce energy through fermentation. A lot of the microbes in our gut, in our intestines, are called obligate or facultative anaerobes. Excuse me. Microaerophiles are organisms that can survive low oxygen environments, so they're not an obligate aerobe. Capnophiles, now those are ones that grow well in high carbon dioxide environments. And I don't have a picture for this, but if you want to grow something in that type of environment, you can, there are kits you can buy that oxidize a reaction that produces carbon dioxide, or you can get a jar. You can put a small candle in the jar, put the plate you want to grow in there, put the lid on it. At some point, the candle will burn up all the oxygen, and in the process will produce carbon dioxide, and you can grow those that way. All right, temperature. Psychrophiles. Those are ones, they're called cold-loving. What they mean is they can actually survive. They can survive low-temperature environments. It doesn't mean they love it. 15 degrees Celsius is not that cold. 20 degrees Celsius is body temperature, but 0 Celsius is really cold. So that would be like 32 degrees Fahrenheit. Mesophiles, they prefer body temp type temperatures. They're usually the ones that make us sick. They're mesophiles. They grow best at moderate temperature. Thermophiles can survive high temperatures. 45 degrees Celsius is pretty hot. That's 100 plus 15 degrees Fahrenheit, really hot. pH, there are organisms that can grow and survive pH. Campylobacter is one of those that causes ulcers, or Helicobacter pylori. H. pylori can buffer acid, so it can grow in low pH. But a lot of times acidity or alkalinity on a pH scale, acidic basic, really affects how organisms grow. Usually it kills them. Acidophiles, and that probably is more of the H. pylori I was talking about. They can survive acidic environments. Okay, water, we did cover in a previous video about osmotic pressure. There are organisms that can survive high osmotic pressure. So in this example, this is actually a red blood cell. This is in the lesson, but this is a hypotonic solution. It's making the cells swell up. This is a hypertonic. See all the solids? And it's causing the cell to shrink. And then this is an isotonic. So actually, the category, there are some, they're called halophiles. They're obligate Halophiles. That means they must grow in high osmotic pressure. Staphylococcus is like that. It grows really well in high salt environments. But this part mentions here, yeah, I was talking about this in the other video. Anything that has any food or a process that has lots of salt and sugar, it will draw water out of cells, and as a result, food will last longer. Like that. Microbes can't survive that. However, like everything else, there are exceptions. This is one of those, an obligate halophile. All right, last thing, and I'm going to summarize this very quickly. And humans have the same thing, what is called a growth curve. And we're talking about microbiology, so here's going to be our example. If you're walking around the building and you swab something, a keyboard, the floor, something that has well, microbes are everywhere. If you take a swab and you take a sample of microbes, you come back and you streak that sample on an agar plate. There's going to be a time, those cells are going to go through a growth curve, and there'll be a time where there's no movement. And that's what this is kind of just referring to. Generation time is the time it takes a cell to undergo mitosis. And like it says here, E. coli takes about 20 minutes or less. Doesn't take very long. And that's one of the reasons you can't leave food out at room temperature for long periods, because if you have 1 million cells per gram, if you leave it out for 3 or 4 hours, you're going to have millions of cells per gram. So everything is written verbatim for you here. This is a great diagram. These are all the parts of a growth curve. The lag phase is where there's little to no cell division, and what usually happens with a microbe or something, if you put it in a different environment, they have to synthesize enzymes, different kinds of enzymes, to break down food in that area. So the lag phase refers to a time where there's really no increase in numbers. Log is exponential growth. That's where the enzymes have been acquired and now you have cell division, so the numbers are going to increase. At some point, if you have a microbe in an area for enough time, they're going to start running out of food and water. So the stationary phase is characterized by cell death equaling the amount of cells dividing. Here, cells dividing is way passing cell death. And if nothing changes, the death or decline phase is where the death of cells is going to surpass the cells dividing. And everything kind of follows that curve. Human beings would. If you put humans on an island, there's going to be a lag phase where they have to adjust to building shelter and finding food, and they might, depending on who they're marooned with, you might have an increase in family size. If you start running out of food, there's going to be a phase where there's no more growth, and then of course if you don't find food, you're going to have people dying. So those are all of the parts of what's called, now we're talking about microbes here, so microbial population growth. But all of them follow this, and you can actually do a science experiment in lab with this. It takes about 5 hours, but you put some E. coli in a broth tube. You then start measuring with a little photospectrometer machine. You put the test tube in a machine, and it shines light through the broth, and it will measure how dense it's getting. And E. coli divides every 20 minutes, so it takes about 4 hours, but you can log this entire curve. And if you leave bacteria in a broth tube long enough, they're going to run out of oxygen. And they're going to run out of food, and you're going to have a buildup of waste, and all the cells will start dying. So, all right. I hope this video helps. Thanks for joining me for this.
And that's the bottom line, because Stone Cold said so.